There's an enzyme folding a molecule in half right now, like it's making a tiny piece of origami out of something that was supposed to kill it. It's not angry, it's not clever, not really, it's just doing its job. Snap, fold, done. And somewhere upstream of that enzyme, a fever is climbing past 104 degrees, and somebody's hands won't stop shaking, and somebody's heart is doing something it's never done before. You're inside a bloodstream that stopped being a peaceful commute and started being a war zone nobody sent an invitation for. It's hot in here. Everything smells like antiseptic and adrenaline. Alarms are going off. Not metaphorical ones, actual hospital ones, the beeping kind. And somewhere close by, a bag of the strongest antibiotic they had left is dripping into a vein like a last resort, because it is one. Yeah, you're probably wondering how you ended up here. Well, you didn't. I did. Oh, hey, sorry. Let me back up. I'm dead. Have been for a while now. Long enough that a while stopped meaning anything because time gets strange once you're not the one spending it anymore. I don't have a body, no pulse to check, no fever to run. What I have is an obscene amount of downtime and a front row seat to watching several billion humans operate machines they were never handed a manual for. I want to be clear about something up front. I'm not a doctor, never was. Credentials feel a little beside the point once you've watched the same biological event happen to different people wearing different faces millions of times over. I don't know things because I studied them. I know things because I watched them happen so often that eventually, even I, a fairly self-absorbed ghost, started taking notes. And this one, the fever, the last resort bag of medicine, the whole unglamorous production, this one's personal. This is how I ended up on this side of the conversation. So, let's talk about what antibiotic resistance is actually doing inside a body. Inside my old one, for a start. And statistically, eventually, inside yours too, unless you get lucky or unless you listen carefully for the next little while. Which brings us to the deeply unglamorous truth about how I got here. It started with a rose bush. Not a car crash, not some ill-advised tropical expedition, a rose bush in my own backyard on an ordinary afternoon, doing the single most mundane task available to a human being, pruning a plant I didn't even particularly like. A thorn caught the back of my hand. I said a word I won't repeat, ran it underwater, called it handled, and went back inside to finish dinner. That's it. That's the whole origin story. If you're waiting for a twist, there isn't one yet. That's sort of the point. Here's the part nobody tells you about your skin. It's not just a wrapper, it's a wall. And once that wall has a hole in it, even a small one, even a rose thorn-sized one, you've opened a door to an enormous population of opportunistic tenants who were, up until that exact moment, none of your concern. Most of the time, that door closes itself. Your immune system shows up, does some quiet cleanup, and you never think about it again. That's what I expected. That's what almost everyone expects every time because it's what usually happens. This time, it wasn't what happened, and the reason why is the entire subject of tonight's little tour. Here's the part nobody tells you about antibiotics. They don't heal you. I know that sounds insane coming from someone who died because one didn't work fast enough, but stay with me. An antibiotic doesn't repair anything. It doesn't boost your immune system. It doesn't stitch your skin back together. It does nothing to your own cells. What it does, the only thing it ever does, is exploit a structural difference between your cells and a bacterium's cells. It's less medicine and more like an assassin who only recognizes one specific face in a crowded room. Take the classic beta-lactam style drugs. Bacteria build a rigid outer wall to keep from bursting under their own internal pressure. Picture a bacterium wearing a suit of armor because the water pressure inside it is constantly trying to make it explode. Beta-lactams jam the machinery that builds that wall. No wall, no armor. And the bacterium's own internal pressure finishes the job on its own. It doesn't get poisoned, it gets popped. Your cells don't have that kind of wall. Structurally speaking, you're a water balloon with better manners, and no beta-lactam has any idea what to do with you. That's not an accident, it's the whole design principle. A good antibiotic is a key built for a lock bacteria have, and you don't. Here's a lock that's even stranger. Sulfonamide drugs. Bacteria have to manufacture their own folate, a vitamin your body just eats from scratch, using their own internal factory. You don't have that factory. You get folate from spinach and cereal and mild guilt about not eating more spinach. A drug that jams the bacterial folate factory does nothing to you because you never had the factory to begin with. It's medicine by process of elimination. Find the one thing only they do and only they need and break exactly that. The word for all of this, incidentally, antibiotic, was coined around 1941 by a microbiologist named Selman Waksman, who went on to personally discover more than 20 of the things himself. It means, roughly, against life, which is an unsettling name once you sit with it, because these compounds were never designed as medicine first. They were weapons first, built by other microbes against each other for a few 100 million years before we ever showed up to borrow the technology.
Which brings us to something considerably older and considerably stranger than anything happening in a hospital. Here's where biology stops being a story about human cleverness and starts being a story about how late we actually arrived to our own show. The instinctive assumption is that we invented antibiotics, bacteria eventually caught up, and resistance is basically our fault, a consequence we brought entirely on ourselves through overprescribing and impatience. And look, we absolutely made things worse. We'll get to exactly how. But we didn't start this war. We walked into the middle one that had been running since roughly forever. Most antibiotics were never invented by a human in a lab coat. They were discovered, pulled out of soil fungi and soil bacteria that had already been manufacturing them as chemical weapons against their microbial neighbors, because the ground beneath your feet is a battlefield roughly the size of the planet, fought at a scale too small to see for stakes that have never once stopped mattering to the things doing the fighting. And where there's a chemical weapon, evolution tends to eventually produce a chemical answer to it. In 2011, a researcher named Vanessa DaCosta and her colleagues published a study in the journal Nature after analyzing DNA extracted from roughly 30,000-year-old permafrost sediment in Siberia. Buried in that ancient ice, they found functional genes for resisting beta-lactam, tetracycline, and glycopeptide antibiotics, chemical classes we wouldn't get around to inventing clinically for another 29,000-odd years. I'll be honest with you here, because honesty is one of exactly two things I have left to offer. Ancient DNA is a genuinely difficult thing to study cleanly. Other researchers in the field have raised fair concerns about this kind of work: contamination from modern sources, small sample sizes, results that don't always reproduce cleanly between labs. That's a legitimate critique, and a responsible ghost mentions it. But here's the part that's much harder to argue with, because nobody had to trust fragile ancient DNA to get it. In 2012, a researcher named Kirandeep Buller and her team went looking somewhere even stranger: Lechuguilla Cave in New Mexico, a chamber system sealed off from the surface world for somewhere between 4 and 7 million years. Not metaphorically isolated, geologically cut off, the same way a sealed jar in your pantry is cut off from this conversation. They cultured live bacteria out of that cave, actual living organisms, not fragments of old code. And those bacteria were resistant, not to one antibiotic, to 14 different modern antibiotics, including daptomycin, a drug doctors keep in reserve specifically for infections that have already beaten everything else. A 2016 follow-up study dug even deeper into one particular cave strain and found 18 separate resistance mechanisms, several of which medicine had never documented in any organism anywhere before. Sit with that for a second. Bacteria that had never once met a human, a hospital, or a pharmacy were already fully equipped to shrug off some of our most serious, most modern chemical weapons. We didn't teach them that trick. They'd had it in a drawer for millions of years waiting for a use case. Evolution didn't build resistance to stop us. It built resistance to stop each other, ages before we existed to take it personally. Speaking of taking things personally, let's go back to the rosebush, because the next 6 days are, in hindsight, a masterclass in exactly how this goes. Day 1, a scratch, some antiseptic, forgotten by dinner. Day 2, a little redness around the cut, nothing dramatic, the kind of thing you notice and immediately decide not to think about because thinking about it feels like overreacting. Day 3, the redness had opinions now. It had spread, it was warm to the touch, and there was a dull, low ache that hadn't been there before. I did what a shocking number of humans do at this exact juncture. I waited one more day to see if it would just go away on its own. Day 4, it had not gone away on its own. I went to a clinic, a doctor took one look, prescribed a standard oral antibiotic, the reasonable, correct, textbook first move, and sent me home with instructions and a completely justified assumption that this would be the end of the story. Day 5. Fever. Not put on a sweater fever, a genuine, unmistakable something is wrong fever, climbing past what any oral pill was going to fix from the outside. Day 6, an emergency room, a blood culture, and a phrase I will never forget hearing out loud, because it's the phrase every infectious disease doctor dreads saying, "It's not responding." The bacteria that had gotten into that scratch weren't just ordinary bacteria having an ordinary bad week. They were carrying resistance mechanisms that made the first-line drug about as threatening to them as a strongly worded letter. So treatment escalated. A second antibiotic, then a third, moving up what doctors sometimes call the ladder. Each rung a drug held in slightly more reserve than the one before it, because every time you reach for a bigger gun, you're spending down a resource nobody's making more of quickly. By the time they found something that worked even partially, the infection had made it into my bloodstream, a condition called sepsis, which is your entire body's inflammatory response spiraling in a way that starts damaging organs that had nothing to do with the original scratch. Sepsis doesn't politely wait for you to notice it. It moves in hours, not days.
I want to be honest about something here, because scientific honesty was the whole deal I made with myself before agreeing to narrate this. I don't know with certainty exactly which resistance mechanism was running the show inside me. Nobody sat me down with a genome printout. What I know is the pattern: a common bacterium, a common first antibiotic, an uncommonly bad outcome. And that pattern plays out in emergency rooms constantly, for reasons we're about to break down properly. Quick round before we go deeper. No lecture, just ammunition. Antibiotics do absolutely nothing to viruses. Zero. A cold, the flu, most sore throats. Pharmacologically speaking, taking an antibiotic for those is like trying to fix a leaky faucet with a legal document. It might feel productive, it is not. Your gut is home to trillions of bacteria that, on a normal day, are doing you actual favors: digestion, vitamin production, keeping troublemakers in line. A single course of antibiotics can take months to fully recover from, and some of those microbial residents may never come back to their old numbers at all. MRSA, the resistant staph bacterium everyone's heard of, isn't some exotic invader. Ordinary staph lives harmlessly on a huge share of healthy people's skin and noses at any given moment. It becomes MRSA the instant it picks up a single mobile piece of genetic hardware carrying one gene. One gene, and the rules of engagement change completely. And in the earliest days of methicillin resistance, the resistant strains had a genuinely bizarre quirk. Some of them only reliably showed their resistance at a lower lab temperature than hospitals were routinely testing at, which means for a while, the very tests designed to catch it were looking in a room that was technically the wrong temperature to see it. Okay, deep breath, back to the mechanics. If an antibiotic is a key built for one specific lock, bacteria have developed roughly 4 different ways to make sure that key never turns. The first is the bouncer. Certain bacteria build molecular pumps in their outer membrane called efflux pumps, whose entire job is to grab anything that looks like a threat and physically eject it back out the door before it can do damage. A nightclub bouncer who doesn't ask questions, just removes you. The second is the editor, or maybe the world's rudest doorman. Some bacteria produce enzymes, most famously beta-lactamases, that intercept the antibiotic and simply cut it apart before it ever reaches its target. Remember that key-shaped molecule from earlier? A beta-lactamase enzyme just snaps it in half mid-air and hands you back the pieces. It doesn't fight the key, it edits it out of existence. The third is changing the lock itself. This is exactly what happened with MRSA. The bacterium swapped its normal cell wall building protein for a slightly different version, one the drug no longer recognizes. Same basic function, different shape, and the drug simply can't grip on. Evolution didn't invent a new lock, it just filed down the one it already had. And the fourth one is the strangest one, and it isn't really resistance at all. It's closer to a magic trick. Some bacteria, a small fraction of any population, essentially play dead. In 1942, a researcher named Gladys Hobby noticed that a small fraction of bacteria, somewhere around 1%, simply refused to die when exposed to penicillin, no matter the dose. 2 years later, in 1944, a physician named Joseph Bigger followed up in the Lancet with the crucial twist. He took those survivors, grew them back into a fresh culture, and exposed that fresh culture to penicillin all over again. If they'd been genetically resistant, the whole new culture should have shrugged the drug off. It didn't. The same 1%-ish sliver survived, and the rest died right on schedule, exactly like the original batch. Whatever this was, it wasn't inheritance. Bigger called the survivors persisters, and the name stuck. Most antibiotics only work on cells that are actively doing something, building walls, making proteins, replicating. A persister cell simply shuts nearly everything down and waits. There's nothing active left for the drug to break. Genetically, these cells are complete identical twins of the bacteria the antibiotic just killed. They didn't do anything clever, they just happened to be asleep when the raid happened, and once the drug clears out, they wake back up and pick up exactly where they left off, part of why some infections seem to vanish and then infuriatingly come back. And where do persister cells thrive best? Biofilms, dense, slimy, highly organized bacterial communities that behave less like a crowd and more like a fortified town. The outer layer is active and exposed. The deeper layers are starved, slow-growing, and packed with exactly the kind of dormant cells that make persistence possible. It's not one trick, it's an entire defensive architecture, built by cells too small to see, engineering a bunker for their sleepier neighbors. 4 tricks: a bouncer, an editor, a locksmith, and a ghost. None of them require malice. None of them require intelligence. They just require time, numbers, and enough exposure to a threat to make the trick worth keeping. Here's the part that should be more famous than it is. We tend to talk about antibiotic resistance like it's a modern scandal, something we did to ourselves through impatience and overprescription, a moral failing with a graph attached. And again, we made things faster and worse, no argument there. But that cave I mentioned earlier, Lechuguilla, sealed off from the surface for millions of years, deserves one more moment because it quietly demolishes the idea that resistance is purely a them responding to us story. Those bacteria never met a human, never met a hospital, a farm, a pharmaceutical plant, or a single dose of anything we've ever manufactured. And they were still carrying, in their own native genetic toolkit, functional defenses against more than a dozen drugs we consider serious, modern, last resort medicine.
That's not a bacterium that learned a trick from us. That's a bacterium that already knew the trick on the off chance it would someday need it, because somewhere in that same underground ecosystem, some other microbe was almost certainly still running the same chemical warfare campaign it's been running for millions of years. We like origin stories where humans are the main character. This one, respectfully, doesn't have us in it until the very last page. And that's half the story, because while all of this is happening at the level of enzymes and cell walls, your brain is running its own, completely separate reaction, one most people have felt and almost nobody's had explained to them. When your immune system engages a serious infection, it releases signaling molecules called cytokines, chemical memos essentially, dispatched to coordinate the response. Some of those memos reach your brain, partly by traveling up the vagus nerve and partly through a few specialized brain regions that are, on purpose, slightly leakier than the rest of the blood-brain barrier. And your brain, upon reading them, does something that feels like betrayal, but is closer to triage. It makes you exhausted. It kills your appetite. It makes you want to lie very still and see absolutely no one. This cluster of symptoms has an actual name in the research: sickness behavior. And the leading explanation, well supported, though not the kind of thing you can prove with a single clean experiment on a human, is that it's adaptive. Your brain is rerouting energy away from things like socializing and digestion and toward the expensive business of fighting an infection. You're not falling apart, you're being redirected. Fever works the same way. It's very likely not your thermostat malfunctioning, it's a deliberate ruse, making your internal environment measurably worse for bacterial replication while giving certain immune cells a performance boost. Reflexively overriding it every single time may not always be doing you the favor it feels like it's doing. And in severe cases, the kind of case I eventually became, infection can produce genuine confusion and delirium, something clinicians call sepsis-associated encephalopathy. The exact mechanisms behind it are still being pieced together, but the practical human effect is almost unbearably ironic. The exact organ you'd need to recognize something is critically wrong is frequently one of the first things a critical infection compromises. Near the very end, I remember feeling less like I was dying and more like I was extremely tired and mildly confused about why everyone looked so worried. That gap between how bad things actually were and how bad they felt might be the single most dangerous symptom nobody puts on a checklist. If you're getting something out of this so far, this is usually the point in the video where I'd ask you to subscribe. So mildly from the void, subscribe. Not because I need the validation. I'm dead. I need almost nothing. But there's more of this if you want it. And unlike me, you still have time to actually use it. All right, feelings and fevers aside, let's talk about scale, because this stopped being a personal anecdote a long time ago. In 2022, 2022, a massive research collaboration called the GRAM Project, the Global Research on Antimicrobial Resistance Project, led by Christopher Murray and colleagues, published the most comprehensive analysis of antibiotic resistance's death toll ever assembled in the Lancet. Their number for 2019, 1.27 million deaths directly caused by drug-resistant bacterial infections, with resistant bacteria playing some contributing role in another 5.95 million deaths on top of that. Naked numbers are useless, so here's the comparison. That direct figure alone was higher that year than global deaths from HIV/AIDS or malaria, two diseases most people would immediately name if you asked them to list humanity's biggest infectious threats. Antibiotic resistance rarely makes that list. It's already earned a spot on it. A follow-up analysis published in 2024 tracked the trend from 1990 through 2021 and found something genuinely unexpected buried in the demographics. AMR deaths among children under 5 fell by roughly half over that period. Real measurable progress, mostly from better general health care. Meanwhile, AMR deaths among people 70 and older rose by more than 80% over the same stretch. This isn't one uniform crisis. It's a crisis quietly changing who it targets, getting better at protecting the very young while getting considerably worse at protecting the very old. That same 2024 analysis forecasts more than 39 million deaths from antibiotic-resistant infections between 2022 and 2050 if nothing meaningfully changes. Though, to be fair, the same researchers estimate that roughly 92 million lives could be saved over that same window with better healthcare access and more careful antibiotic use. The bad number and the hopeful number came out of the same model. Which one we get is genuinely still up for grabs. You've probably also heard a different, scarier number. 10 million deaths a year by 2050. That figure comes from a 2016 UK government-commissioned review led by the economist Jim O'Neill, and it's the one that ended up in headlines. But it's worth knowing that other researchers, Marlika de Cracker and colleagues in a 2016 PLOS Medicine paper specifically, publicly questioned the methodology behind that particular projection, arguing the data available at the time couldn't really support a number that precise. I'm telling you this not to let anyone off the hook, because the honest version of this story includes scientists disagreeing with each other in public, which is generally a sign the process is working, not a sign the threat isn't real. Let's clear out some things you almost certainly believe, because I believed most of them too, right up until I had extremely personal reasons to look closer. Myth 1. You become resistant to antibiotics. Reality? You don't. Bacteria do. Your body doesn't build up a tolerance to penicillin the way it might build a tolerance to caffeine. What actually happens is that overuse gives more bacteria more chances to develop and share resistance mechanisms among themselves. Why it's false? The confusion is understandable, but it's backwards. The pill isn't losing its grip on you, it's losing its grip on them.
Myth 2. You must always finish the entire course no matter what, or you'll create resistant bacteria. Reality, this one's genuinely more complicated than the poster in your doctor's waiting room suggests. A 2017 analysis in the BMJ, led by Martin Llewellyn, reviewed the evidence behind that advice for common infections and didn't find solid support for the idea that stopping early causes resistance, while flagging real evidence that taking antibiotics for longer than medically necessary can increase resistance risk instead. Why it's false, or at least oversimplified? Duration isn't a universal rule of thumb. It's a decision that depends on the specific infection and drug, which is exactly why the honest advice is follow your own doctor's specific instructions, not a slogan off a poster. Myth 3. Antibiotic resistance is a hospital problem, and hospitals are someone else's problem. Reality, resistant bacteria show up in community infections, in food, and in water. And antibiotic use in livestock and agriculture is a major driver of resistance, entirely separate from anything a hospital does. Why it's false? The bacteria don't check your zip code before deciding where to evolve. Myth 4. Don't worry, scientists will just invent new antibiotics to keep up. Reality, the great wave of antibiotic discovery mostly happened between the 1940s and the 1960s, and it has slowed dramatically since. Genuinely new classes of antibiotics are rare, and the economics are brutal, because a good new antibiotic is one you deliberately use as little as possible to keep it working, which is a strange business model compared to a drug people take every day for years. Why it's false? They'll figure it out assumes an industry incentive structure that, right now, mostly isn't there. Here's a piece of good news, immediately followed by the bad news that cancels most of it out. Building resistance usually isn't free. An efflux pump takes energy to run. A modified cell wall protein sometimes works slightly worse at its actual day job.
In a world with no antibiotic present, a resistant bacterium is often, not always but often, at a slight competitive disadvantage next to its unmodified neighbors because it's spending resources defending against a threat that, in that moment, isn't even in the room. That's the good news. Remove the antibiotic, and in theory, resistance should slowly fade, outcompeted by leaner, faster reproducing bacteria that never bothered building the defense. Here's the bad news. Evolution doesn't stop at good enough. Bacteria frequently develop what are called compensatory mutations, additional genetic tweaks that patch up the performance cost of resistance without giving up the resistance itself. Over enough generations, you can end up with a bacterium that's fully resistant and just as fit as one that never bothered, which means removing the antibiotic doesn't reliably make the problem go away. The debt doesn't always get called in. Sometimes the bacterium just refinances it. This is why stop using this drug for a while and resistance will fix itself is a much less reliable strategy than it sounds like it should be. We'd love evolution to be a self-correcting system that rewards patience. Mostly, it rewards whatever survives by whatever means happen to work indefinitely. Quick one. Picture your own medicine cabinet for a second. I'll wait. Somewhere in there, there's a decent chance you've got a half-finished blister pack from an infection you had 18 months ago, kept just in case. You get a scratchy throat next winter, you're busy, a doctor's appointment feels like a lot of effort for what's probably nothing, so you take 2 of the old pills and call it preventative. Here's what you've actually done. Taken an unknown drug at an unknown and almost certainly insufficient dose for an infection nobody confirmed was even bacterial based on symptoms that overlap almost completely with a virus those pills can't touch anyway. If there were any bacteria involved at all, you've just given them exactly the kind of low-dose, incomplete exposure that functions as free evolutionary target practice, precisely the scenario Alexander Fleming himself was worried about out loud in his 1945 Nobel Prize lecture, decades before your cabinet existed. It felt responsible. It felt like taking initiative. Biologically speaking, it was closer to handing bacteria a pop quiz and hoping none of them study. Enough dread. You didn't come here to feel powerless. You came here, whether you knew it or not, to feel slightly more in control of a body you've been operating without a manual your entire life. So here's the actual practical framework. No mysticism, just the stuff that works. Take antibiotics only for what they're actually prescribed for, and only when a doctor has genuinely confirmed or strongly suspects a bacterial infection, not as a just-in-case security blanket. Stop pushing for antibiotics when you've got a cold, the flu, or most sore throats and coughs. It's not that your doctor's being stingy. The drug does nothing to a virus except potentially harm your own microbiome while failing to help you. Take the exact dose at the exact frequency for the exact duration your doctor specifies, and if you're unsure whether you should stop early or keep going, ask them directly, rather than defaulting to a rule you read somewhere once. Never take leftover antibiotics from an old prescription, and never share yours with someone else, no matter how similar the symptoms sound. Dispose of unused antibiotics properly. Many pharmacies will take them back rather than tossing them in the trash or flushing them since they can leach into water systems.
And keep exerting evolutionary pressure long after you've forgotten you ever had them. Get the vaccines recommended for you. Fewer viral infections means fewer secondary bacterial infections and fewer moments where someone's tempted to prescribe an antibiotic just in case. Take basic infection prevention seriously. Hand washing, proper wound care, food safety, because the single best way to avoid needing an antibiotic is to avoid needing one in the first place. Pay a little attention to where your food comes from. Antibiotic use in agriculture is a genuine driver of resistance, and demand for more responsible practices does eventually filter upstream. None of these require heroics. All of them, done consistently by enough people, actually move the numbers from a few minutes ago. Now, before you go looking at your own rose bushes with suspicion, let me reframe something, because fear was never really the point of any of this. Antibiotic resistance isn't a monster. It isn't malicious, it isn't personal, and it isn't even particularly clever in the way we usually mean that word. It's just natural selection running at a scale and speed that's honestly kind of magnificent once you stop being afraid of it. A population of organisms too small to see, solving a chemistry problem with sheer numbers and time. The same blind, patient process that built every eye, wing, and immune system on the planet. The fear is fair. I'm not going to pretend I found any of this soothing while it was actively killing me. But fear without understanding just sits there, useless, taking up space. Fear with understanding turns into something closer to respect for your immune system, which wins this fight the overwhelming majority of the time without you ever knowing it happened, for the drugs, which are borrowed weapons from an ancient war on loan to you that still work more often than not, and strangely, even for the bacteria, who aren't cheating, they're just very, very good at their one job. You're not supposed to walk away from this terrified of your own garden. You're supposed to walk away slightly more equipped, which statistically makes it considerably more likely you get to keep having ordinary days, boring days, the kind where a scratch is just a scratch and dinner gets made on schedule and nobody's taking notes on your bloodstream from the other side. If eternity's taught me anything, and the sample size is generously enormous, it's that almost nobody sees the moment coming where probably nothing quietly turns into actually something. I certainly didn't. I stood in my own kitchen with a bleeding hand and a completely reasonable assumption that biology owed me a routine recovery because it always had before. I wish I'd known that a scratch isn't a small event, it's an open door. I wish I'd known that "it's probably fine" is a sentence worth double-checking after day 3, not day 6. I wish I'd known that finishing my antibiotics wasn't the moral question I thought it was, and that the real question was always whether I needed them in the first place and exactly how my doctor wanted them used. Mostly, I wish I'd understood that my body was never trying to be comfortable. It was trying to keep me alive using tools built over billions of years for exactly that job, and that the version of me arguing with a rosebush was, without realizing it, standing directly in the middle of a war that started long before humans, chemistry, or bad gardening decisions existed. I don't get a body back. That part of the deal is non-negotiable, and frankly, the paperwork on this side is worse than you'd think. But you get to keep yours a little longer, a little smarter, a little more alert to the difference between a scratch and an open door. Go finish your dinner, wash the cut, call the doctor on day 3, not day 6.